Why it's not going? Next slide. It is issue. Next slide. First of all, I would like to start with the housekeeping announcement. This seminar is being recorded. Someone asked in the chat question that yes, we are recording and it will be available later. So we encourage asking questions. Please use the chat panel for question at any time. Participant audio is muted. After the presentation, you can raise your hand by tapping the hand icon as shown in the picture and pop up at the bottom of your screen. We can call you in the order that you raise your hands. We will unmute your microphone when it is your turn to ask question. If you need more information, contact contract manager me, Ninin Aung, at arb.ca.gov. Information about the presentation slide and other material can be found at CARB website. The recording will be made available following today's seminar. Let's get started. First, I will give you a project overview and introduce the speaker. Second, PI will present the research finding. After his presentation, we will have a question and comment period. The project overview is CARB question is defined as what are the health effects of the combined exposure to ultrafine particulate matter and ozone. The importance for CARB is to investigate the combined effect of multi-pollutant exposure, establish biological plausibility for multi-pollutant health effects, and impact future research direction and standard evaluation. So I will introduce today's speaker. Today's speaker is Dr. S. Schrager. Dr. Schrager is also principal investigator of this project. Dr. Schrager received his PhD in physiology from UC Davis. Currently, he is a department chair for anatomy, physiology, and cell biology in the School of Veterinary Medicine, UC Davis. His expertise is in pulmonary neurophysiology and how environmental factors affect human health. Additionally, Dr. Schrager has authored multiple publications examining factors that contribute to the exposure response of ozone in humans. His finding of the ozone-induced adverse health effects in human study are central to the U.S. Environmental Protection Agency 2009 Integrated Science Assessment Proposal to lower the national air quality standard for ozone from 75 parts per billion to 70 parts per billion. Now, Dr. Schrager will present the research finding. So take it away, Dr. Schrager. So I will stop sharing my slide.
All right.
Thanks.
I will try to start sharing my slides. How about that?
Yes. Yes. Thank you.
All right. Share.
Yes.
All right. All right. Thank you for the introduction. This is I'm going to be reporting on data that we collected in the most current CARB contract that we had, specifically looking at tissue that was collected in another study previously. But before I get started in that, I just wanted to kind of talk a little bit about, since I have the opportunity here, this is interesting to me. I don't know, many of you know me, but I'm about to retire. And my very first support for research came from the Air Resources Board, and actually supported my PhD work at UC Davis under the direction of Bill Adams. So I kind of go way back with the Air Resources Board, and I've always appreciated working with the board and you know, the whole process of that research. So to get started, we're talking about particulate matter, and in this case, ultrafine particulate matter. And in the context of this study, we were interested in the cardiorespiratory or cardiovascular effects as overall. As far as, let me make sure I've got my laser pointer working. All right. There's multiple epidemiologic associations that have been made between PM of different sizes and cardiac events, mortality, arrhythmias, myocardial infarction, stroke, heart failure, have all been observed in epidemiological studies. Takeaways from those studies would be that there's a higher incidence of PM-related cardiac events in mature populations with pre-existing cardiovascular disease. So individuals already have some degree of cardiovascular disease present, and usually that's an elderly population. Exposures to heavily trafficked roadways or proximity to heavily trafficked roadways can be a risk factor. And in that case, when we start looking at that pollutant or that combined pollutant environment, we're talking about PM size that's relatively small, usually an ultrafine particle, and a very, diverse composition. And then associated with that particulate exposure in that environment is also gaseous pollutants, and the one that we were concerned with was ozone in this case. So what we wanted to do was essentially use an animal model where we could look at not only ultrafine particulate matter exposure alone, ozone alone, but also the two in combination and combine them. And so there have been studies that have looked at both PM and ozone exposures in rats with some that have genetically, or have a propensity or risk for developing cardiovascular disease. However, most of these studies, matter of fact, at the time of that we started this project, there were no studies that looked at elderly or mature rodents. Most of the studies were done in adolescent animals with or without genetically induced hypertension. And so when we look at these models for hypertension and you look at the progression of cardiovascular disease in them, there is actually a progression that you really have to get out to past 40 weeks to actually start seeing some of the marked changes in remodeling of the tissue in the heart that would be consistent with chronic disease. So what we decided to do is essentially follow up on that lack of information and actually use a model of mature animals, about a year old. They were retired breeders, to examine this question. And really, what are the effects of exposure to ultrafine particulate matter, ozone, and the two combined? And we use spontaneously hypertensive rats, mature adult, which if you look at the literature means that they're about a year old or older. And they have, or have been shown to have pre-existing cardiovascular disease, which I'll get into a little bit how we characterize that. And so specifically in this study, what we did is we took banked lung and heart tissues that were collected previously in Fern Tablin's CARB contract, which I was a co-PI on, which was titled Co-Exposure to PM and Ozone, Pulmonary C Fiber and Platelet Activation in Decreased Heart Rate Variability. So the title is, I think we were thinking what was going to be happening here, and what we found was slightly different from what the title might indicate. So let's get into it so you actually know exactly what we found. What I want to do is talk about the research design for that original study, because that's where the tissues came from for this study, and kind of give you an overview of the high points of that data. so that you understand why we felt it would be worthwhile examining tissues, both from the lung and the heart, from that study. So we had a rodent model of cardiovascular disease, spontaneously hypertensive rats. These are a genetic variant of the Wistar Kyoto rat. And we used the Wistar Kyoto rat as a control group in this study. So we had both spontaneously hypertensive rats as well as Wistar Kyoto rats as our controls. The animals were approximately 50 weeks of age. These were retired breeders, so they were all males. And studies have shown that at this age, there's considerable cardiac remodeling in these animals with left ventricular hypertrophy and fibrosis of the myocardium. And they demonstrate increases in spontaneous arrhythmias, including atrial fibrillation, spontaneous atrial fibrillation, AV block, and PVCs. So they show a lot of symptoms or characteristics of a population that you would see in humans, of an aging population with chronic hypertension and heart disease. So what we did as far as our pollutants, as I said, we had filtered air as our control. We had ultrafine particulate matter, which simulated traffic-related exposure. It was a pre-mixed flame-generated ultrafine particulate, around 70 nanometers in size. And the concentration we used was 250 micrograms per cubic meter. We also had an ozone exposure branch here, or arm to the study, which was to 1 ppm. And then we had the combined exposure to these two pollutants combined. The exposure protocol was 1 hour of baseline, so the animals were put into the chamber for 1 hour prior to the beginning of exposure. We had a 6-hour exposure followed by an 8-hour recovery period. Prior to exposure, 2 weeks prior to exposure, or more 2 weeks, the animals were instrumentated with telemeters. ECG telemeters were implanted for the measurement of heart rate and the evaluation of arrhythmias, and eventually the study of heart rate variability. We also collected blood post recovery. So at the end of the study, for analyzing systemic inflammation and platelet activation and changes in platelets. We also looked at fixed the lungs with 10% formalin at 20 centimeters of water. So we did an air fixation, and we fixed the hearts also in 10% formalin. And then we examined the lungs histologically, and in this study, we also did that with the hearts as well. And so let's now look at what we saw. First of all, how do we examine and analyze this data? Because the data was not normally distributed, we ended up using non-parametric statistics, Kruskal-Wallis test, and with Bonferroni post-hoc test comparisons were used to distinguish by strain. So was there a difference between the strains, and also strain-related differences. So distinguish by strain as also strain-related differences using a Mann-Whitney test, which was also a non-parametric test. P-values for significance was set at 0.05. So to get started talking about the actual data, one of the things that we did, obviously, we took ultrafine particulate matter, we generated it. It was characterized, we characterized it both for its size and its concentration, but also for its content and the polyaromatic hydrocarbons that were contained within it. And the ultrafine particulate matter by itself had a lot of naphthalene derivatives in it, fluorine as well, and some derivatives of biphenyl. And so if you look at that, the red indicates components that were also seen within what do you call it? Within these are all of these are vapor within the vapor component of the atmosphere, and the P indicates that was associated with the particulate itself. Now, we did combine this with ozone. Now, one of the important questions that we had right off the bat, are we changing the composition of our particle by combining it with ozone? And indeed, our data would indicate exactly that. We still had our biphenyls here, but almost no, except for the 2-ethyl naphthalene down here, no naphthalene or naphthalene products. And so we also only saw the derivative of certain things that also began to appear, pyrene, anthrone, all these multiple ring hydrocarbons that are relatively large. So they were both seen in the, in the vapor phase as well as the particulate phase of this exposure. So we were changing the composition of our particulate by combining it with ozone. So the question became, is it appropriate to do a direct comparison between these two? And so we kind of in the current study, we have broken out UFPM compared to filtered air, and then UFPM plus ozone compared to ozone and filtered air in our analysis. One of the things that is important in this initial study that we did was we saw a clear difference between the Wistar rats, or our baseline animals, and the response to ozone compared to the spontaneous hypertensive rats, or SH rats. And so when we just exposed them to ozone, there was a marked and significant differences between strain, indicating in the lung that the spontaneously hypertensive rat, for whatever reason, was more sensitive to the oxidant stress of ozone by itself. And if you look at the differences, they're marked, for some of these markers, clearly there's still some epithelial necrosis in the terminal bronchioles here in the Wistar rats, but it's much more severe in the spontaneous hypertensive rat. PM, neutrophil infiltrate, again, significantly higher. And then markers like large airway ciliary loss, markedly greater in the spontaneously hypertensive rat. It's really not present in the Wistars. And exudate was also observed in the spontaneous hypertensive rats, while it was not present in the Wistar rat. So again, a higher sensitivity to the oxidant stress associated with ozone. So that's almost like we could consider that as a characteristic of our spontaneously hypertensive rat in this study. The other thing that we saw, which was also interesting because it goes further to characterize our model here, is that when we looked across exposures and we were looking at the hearts, we observed focal organized necrotic lesions, which are in this box. An example is in this box. And these were elevated across all exposures. Really, there was no significant difference across, but what was significant was that when we compared across strain in every exposure, there was a significant increase in these lesions, which tend to say that there was something associated with the strain itself and chronic injury. And so this would be a marker of chronic injury that is ongoing in this model, and whether that's associated with the hypertension that's present or not is not clear, but clearly this would be a marker of injury, chronic injury of these animals that was present prior to exposure. Now, one of the critical observations from the previous study was the observation of acute injury. So acute myocardial necrosis, that's indicated by these vacuolized cells here, lysosomes in these cells, indicating acute injury and acute necrosis. And we observed that both with the ultrafine particle by itself, and it was greater than across strain, so greater in the spontaneously hypertensive rat compared to the Wistar rat. There was, some effects here with the ultrafine particles in the Wistars, and again, much greater effects in the combined exposure cross strain. So we have an effect of this combined pollutant as well as the particle by themselves causing these lesions, which are injury to the myocardium. So what was really drove the current study, the new data that I'm going to be talking about, was the question: what are the factors that contribute to this ultrafine particle by itself and ultrafine particle plus ozone induced acute myocardial injury? What are the factors that lead to this injury? Because this was the first observation that we could see of a pollutant-induced injury to the myocardium. So what we did is we went and said, okay, within the previous study, we looked at our injury within the lung. I showed you the data for the exposure by the ozone exposure, but when we looked within the lung itself, we saw that indeed there was more injury with the combined exposure. There was almost a very low injury with ozone, but in the combined exposure, there was marked increase in injury that was greater not only than filtered air, but also greater than the particulate exposure by itself, the ultrafine particulate exposure by itself, or the ozone by itself in the Wistar rats, as well as in the spontaneously hypertensive rat as well. So where we also had injury with ozone. So we have injury and increased injury associated with combining the ultrafine particulate matter with ozone in the lung. Now, I also said, and this kind of contributes to this question of, well, what really contributed to this injury in the myocardium? So we did go back, we looked at heart rate variability and arrhythmias, and what we found was that in the ozone and in the ultrafine particulate matter plus ozone atmospheres, there was an increase in and significant alterations in heart rate variability parameters, cardiac arrhythmias, both in the spontaneously hypertensive rats, but also in the Wistar rats as well. And what we saw was that there was an increase in NN interval. Essentially, that's the inverse of the time between heartbeats. So what this means, if it goes up, that means heart rate is decreasing. There's a significant decrease in heart rate. This is a marker of essentially standard deviation of the variance in heart rate, and specifically it's normalized. So it's a good marker of parasympathetic outflow to the heart. There's another marker that we looked at, which is high frequency power, which is derived from heart rate variability analysis, frequency dependent heart rate variability analysis. And that was also increased, also a measure of parasympathetic outflow. We also saw, as far as arrhythmias, we saw marked increases in AV block, or atrial ventricular block, so the conduction of the electrical signal between the atria and the ventricles in the heart, in both number and severity. And all of these are caused by increased sympathetic outflow to the heart. We also, though, saw within the data increased low frequency power and an increase in the ratio between low frequency power and high frequency power. We saw increase in PVCs, or premature ventricular contractions, which is an important arrhythmia in heart disease, and decrease in the approximate entropy, which is another measure derived in heart rate variability analysis. And all of these indicate we also have an increased sympathetic outflow. So we're having this co-activation of both this parasympathetic system and the sympathetic system that supplies the heart, leading to all these changes. But the important thing to note here, when we start thinking about what caused that injury, is that this is with ozone and PM plus ozone. So it would seem that these changes are primarily more related to the oxidant stress of the ozone exposure than they are to necessarily the particulate exposure. The other thing that we looked at, and really another thing that drove the question and some of the measures we did in the current study, was that when we looked at the blood and we looked at CD61, which is a measure of platelets, that exposure to ozone and to the ultrafine particulate matter led to a decrease in circulating platelets in the spontaneous hypertensive rats. So the question is, where are these platelets going? Are they forming clots? Are they going into the vasculature in different organs? Where are they going? One of the things that kind of indicates what's happening is that we see an increase in aggregates of platelets interacting with neutrophils as well as leukocytes, and that these are elevated baseline in our spontaneous hypertensive rat, which tends to say these animals are more prothrombotic, more likely to form clots and have a high baseline for doing so. But these circulating thrombi or aggregates disappear with exposure, both to ozone as well as to the PM exposure. Those observations were combined when we looked histologically in the lung with a tendency for there to be an increase compared to filtered air in fibrin neutrophil aggregates within the vasculature. And that's what this indicates. Even though it wasn't consistently significant, it did suggest that there was something at least happening with regard in the tissue with regard to microthrombi being lodged within the vasculature, and this was for the lung. So if we go and we look at what we saw increased platelet activation, we saw parasympathetic sympathetic nervous system co-activation, we saw cardiac arrhythmias that became quite severe in these animals, and we saw myocardial injury in the spontaneously hypertensive rats when exposed to the ultrafine particulate matter by itself and when it was combined with ozone. So what we wanted to do was look at this more closely using the tissue that we had saved from this previous study. So our hypothesis was O3 enhances the biological potency of the particulate matter, promoting more reactive oxygen species production, and at the particle surface, which would potentially increase its toxicity and increase the cellular antioxidant expression within the lung and the tissues with those exposures. Increase oxidant sensitivity of the spontaneously hypertensive rats, i.e. the increased responsiveness to ozone that we saw. Would that make them more susceptible to microthrombi formation and myocardial damage? Again, is there this relationship between the oxidant sensitivity of the spontaneously hypertensive rats and the development of myocardial injury? And so what we wanted to do is look at platelet leukocyte and platelet neutrophil interactions. And we did it in this study by looking at microthrombi or fibrin aggregates within the tissues, both within the lung and within the heart. So that's kind of our rationale. I'm not really going to repeat our rationale, but our approach was to evaluate this initially, and our objective one was to define the effects of O3 on the local relationship between particulate deposition and cellular oxidative stress. So going back to, if we look at the deposition of the particles and we look at the same region of the airway, do we see increased markers of oxidative stress within the airways? So what we did is we attempted to look at the spatial proximity of particles deposited in the airway and markers of oxidant stress and metabolism. So what we did is we took tissue, we double stained it for nerve-dependent phase 2 antioxidants, hemoxygenase 1, so HEMOX1, and superoxide dismutase 1, SOD1, and these were examined using dark field microscopy complemented with a hyperspectral imaging system, which allowed us to visualize where the particles were simultaneously. Now, this is where we had problems in this study, and reporting negative, I think it's important to report negative findings sometimes, and this is, I think, one of them. And we saw no changes when we looked across the lung for our markers of oxidant metabolism, either via our HMOX1 or our SOD1, and across exposure and across strains, we did not see any effects. And then in addition, we had problems visualizing particles and where they were lodged within the airway. Probably what caused this was the fact, one, we used airway fixation, meaning we put our fixative down the lungs, which probably potentially dislodged particles, but also we waited for 8 hours post-exposure. So most likely, most of the particles were either moved up the tracheal, the ciliary escalator in the airways, or absorbed, because they're ultrafine particles, into the tissue and then subsequently into the bloodstream. And it's also, with these markers, it's very likely that we did not hit the correct time point to look at these markers. It would have been better if we would have been able to have tissue that was collected immediately after the end of the exposure to do this study, this component of the study. So objective 2 for our study was to characterize ultrafine particle plus ozone impact on microthrombi formation. And so that goes back to that original data that we saw that indicated that there might be something happening there. And so we looked at platelet activation, based upon our previous observation, platelet activation, so on and so forth. What we did is we looked at fibrin stabilized aggregates, which would be microthrombi, in the lung and myocardial tissue, specifically from the ventricles of the hearts, using a phosphotunsic acid hemoxylin stain, or PTAH, easier to say, to look at these using histology. What we did is then, when we looked at the tissue, we decided that there was kind of a variability in the types of lesions we saw. And so we characterized the types of lesions into 3 categories: category 1, category 2, category 3. And category 1 was a small aggregate with an arterial or artery, so there would not be complete occlusion of the vessel. And these were arterioles or arteries. And category 2 was an aggregate fully occluding arterial. So this would be complete blockage of an arterial. So it would be a small artery. And then category 3 was a large multifocal aggregate, so within an artery, but without complete occlusion. And so, having characterized the lesions, the question is, well, how many of these lesions did we see? So what we did is we counted the number of lesions in each category within the sections that were being observed. And so within a section, 0 would be none, 1 would be 1 to 3, a score of 1 would be 1 to 3 of these in that category. 2 would be 4 to 6, 3, 7 to 9, so on and so forth, all the way up to where we're seeing 13 or more, which would be a score of 5. So using that, and if we look at the lung over here off to the right here first, and we look at the particulate effects here, indeed, when we look at the particulates for category 1, microthrombi as well as we see an increase within the spontaneously hypertensive rats and also within the Wistar rats compared to their controls exposure to filtered air, which would be the white and the green here. So for both strains, there's a significant increase in category 1 and category 2 microthrombi in the lungs of these animals, caused by the UFPM by itself. If we look at the lungs within the, when exposed to ozone and to combined exposure, both for the Wistars and the spontaneously hypertensive rats, again, we see this pattern where we see an increase both in the spontaneously hypertensive rats and the Wistars, but where these differences are significantly, again, difference between the strains. So there's a significant difference between the strains as well as between filtered air and ozone. So again, for category 1 within the lung, specifically, very marked increase in those lesions. Category 2, again, we see an increase in that lesion compared to the normal Wistars as well as compared to ozone and to filtered air. And again, also for category 3. So there's some large actual arteries, larger arterial vessels that show some microthrombi within them as well, within the spontaneously hypertensive rats compared to the Wistar rats. Now, when we look at the hearts, and we go and look at the hearts, and remember that's where we saw our lesion, our acute myocardial necrosis. Question is, could it be possible that we're having local ischemia that is leading to those lesions? And so when we look at into the heart with the UFPM exposure or the ultrafine particulate matter by itself, indeed we see in the spontaneously hypertensive rat an increase compared to the Wistar rat, an increase in category 1, category 2, and category 3 microthrombi lesions for all these categories compared to the Wistar rats. The Wistar rats also had lesions, but only category 1, so partial occlusion of small arterioles or arteries. If we now look at the lesions within the hearts of the spontaneous hypertensive rats exposed to the combined pollutant compared to ozone and filtered air, then what we see is again this marked significant difference within the hearts of these animals compared to exposure, compared to the Wistar rats, as well as within the strain to filtered air and to the ozone by itself. So really there's no significant effect in either filtered air or with ozone by itself. And we see this effect for the difference between strains going through all these categories. So we're seeing also larger lesions in larger arteries, which is an example right here, in our spontaneously hypertensive rats, as well as category 2, again, complete occlusion of our arteriole here. There's another one over here, actually. And then a partial occlusion in a, again, small artery arteriole in this example. So this data is really consistent with what we saw with a decrease in platelets in the circulating blood. Where are they going? What are they doing? And also the indication or the suggestion based upon the platelet white blood cell aggregates that we saw disappear from the circulation, that they're going not only into the lung, but also into the myocardium. Objective 3 of the current study was to define the effects of the combined exposure on myocardial injury. And so for here, what we were doing is we were looking at markers of ischemia, as well as markers of nonapoptotic cell death, because when you look at that lesion with the vacuolization, it suggested similar lesions that we've seen, that we've been able to label those cells with a NER77 marker, and June B we used as our marker of a myocardial ischemia. And this has been used in cardiovascular studies to look at myocardial ischemia in different models of cardiovascular disease. And so also along with this, because we want to further characterize these animals that we used in this study, we also looked at the degree of cardiac fibrosis that was present in our animals in this study, both the Wistars as well as the spontaneously hypertensive rats. And so what we saw here, this is our down here is our markers of injury and ischemia. Again, very mild signal. And again, I think this is related to timing of our study and the time it takes for these markers to appear. And that, I think, was what caused essentially our negative result here. I'm not sure that there wasn't some mark, that there isn't, matter of fact, you have to think there has to be some degree of ischemia with the vascular occlusion that we saw, but I just don't think we're catching a good marker of, or a good time for looking at these markers. However, when we look at fibrosis, which is this data up here, you see a consistent elevation in fibrosis across every exposure compared to every exposure for the Wistar rats. So higher fibrosis in the spontaneous hypertensive rats than the Wistar rats. And this data, we combine the data from the Wistars and the spontaneous hypertensive rats, that's what's shown here, spontaneous hypertensive rats, and the degree of fibrosis is much higher. This is the great scoring grading system we used for evaluating this. And these are examples of the type of fibrosis that you see in this tissue, where you have essentially a dissecting fibrosis, where the spaces between the normal myocardium becomes filled with collagen, or you get this replacement fibrosis where you've had damage to the myocardium and the tissue and the myocardium itself is being replaced by fibrotic tissue and collagen. So this would be considered a severe lesion here, and this would be a more mild lesion associated with fibrosis of the tissue. So consistent with chronic myocardial stress in our model and previous observation of our increased focal necrosis, which was pre-existing prior to our exposure, this is consistent with these mature adult spontaneously hypertensive rats having a baseline cardiovascular disease. So how do we pull all this together? There's always a question when we do this. Our important observation here was the fact that we were seeing vascular occlusion with microthrombi, not only in the lung and arterioles within, pulmonary arteries, within the pulmonary arteries, but also within the, what do you call it? The coronary circulation. And so this kind of confusing. Hopefully I can walk you through this without and kind of make it a little more clear. Essentially, you have compartments, and the first compartment we're dealing with is at the atmospheres, what's happening in the atmospheres. We know that ultrafine particulate matter and ozone interact to produce our changes in our particulates as well as vapor phase. These are going to go into the lung, be inhaled into the lung, interact either with the airway lining fluid, especially ozone, and the reactive and vapor components of the particulate matter, and then eventually to the airway epithelial cells. And with regard to the particulate components, direct interaction with the airway epithelial cells, alveolar epithelial cells. And at that site, we would expect to see reactive oxygen species generation, which has been measured before, which leads to oxidative stress, airway injury, and airway inflammation, all which has been demonstrated with ozone and other oxidant stresses before. And we would have to throw in our combined exposure in this case, where we saw marked increases in the lesion within the lung. Now, as a side note, not done in this study, but goes back to the topic of the previous study, we know this activates lung sensory nerves and eventually causes some central nervous system effects, which is related to the activation of the parasympathetic nervous system and the sympathetic nervous system, and the changes we saw in heart rate variability, and also contribute to the arrhythmias that we saw in the previous study. But important for the current study was that it appears this activation and potentially the movement of the particulate, these being very small particulates, they're being taken up, potentially not only being acting within the airway, but also potentially acting on the epithelium of the systemic circulation, as well as the coronary circulation, which are these, this is the systemic circulation compartment, and this is the myocardium and the associated coronary circulation compartment down here. And in both of those compartments, we would see the activation or the potential further disruption of endothelial function, leading to endothelial dysfunction, platelet activation, and microthrombi formation. And these microthrombi in the systemic circulation could then go to the lung in the pulmonary circulation, the pulmonary arteries, causing the vascular occlusion we saw, and also go to the coronary circulation. But you could have those exact same events occurring in the coronary circulation, and the production of platelet or platelet activation and microthrombi and the resulting vascular occlusion also occurring locally within the myocardium. That vascular occlusion within the myocardium would lead to ischemia and potentially contribute to the injury that we saw in our previous study. Contributing to that injury and ischemia could be another factor, and that's those PVCs, or premature ventricular contractions, are also known to produce ischemia in the myocardium and can further contribute to this injury. In addition, ischemia and injury of the myocardium can send signals via afferents or sensory nerves within the heart itself, and also feed back to the central nervous system and increase sympathetic nervous activity. So we may have a loop, a positive loop here that is being generated here that is further contributing to cardiac dysfunction. So conclusions: exposure to ultrafine particulate matter and ultrafine particulate matter plus ozone promotes the formation of microthrombi in the heart and lung, and mature spontaneously hypertensive rats are more susceptible to the thrombotic effects of air pollution that we used, specifically the ultrafine particulate matter and the combined exposure, ultrafine particulate matter and ozone exposure that we used in this study. And so there are definitely strain-related changes in the platelet white blood cell aggregates consistent with cardiovascular disease. We see that in cardiovascular disease. There's a strain-related increase in oxidant stress and within the lung that we could see. And we could see that there's a strain-related increase in chronic myocardial injury and fibrotic remodeling in our model. So when we look at our model as a model of cardiovascular disease in an elderly population, this is a good model for that. And it makes these animals more susceptible to the thrombotic effects of these exposures. So the bigger conclusion, if we're trying to make some type of translation to the human population, was that it would support that individuals with pre-existing cardiovascular disease are at greater risk for adverse as well as heart as well as lung effects of oxidant and ultrafine particulate pollution, really supporting the notion that this is a susceptible population and should be considered in the standard setting process. So I just wanted to finish off by acknowledging the multiple collaborations and our funding for this study. Emily Wong, who is was a postdoc in my laboratory, did a lot of this work and the lion's share of the work really, and got to hand it to her. She was instrumental in having this study move forward. William Walby, who was critical in actually collecting the original tissue and running the exposures in the previous study. Fern Tablin, of course, who collaborator on the study and continues to provide consultation. Dennis Wilson, who did the initial histological examination of both the heart and the lungs. Ron Lee, who helped with the PTH staining or told us how to do it, gave us advice there. Josh Stern, our cardiologist, who looked at and evaluated for heart for the arrhythmias. Laura Van Winkle, who was very helpful with the hyperspectral imaging and looking at the particles within the tissue or attempting to do that. And her advice is always very valuable when looking at tissue. Dr. Rachel Reeder, who is the head pathologist at the California National Primary Research Center. She was very helpful in looking at and evaluating and setting up the semi-quantitative measures of both the platelet distribution or microthrombi distribution and categories, as well as re-examining the hearts and re-scoring the lesions within the hearts themselves. So, questions?
Thanks, Dr. Schriegel.
Yeah.
Is there any questions from the audience? So please raise your hands if you have questions so that we will call you and unmute your audio. Okay, since we are waiting, I have a question. Oh, there's somebody asking. Thank you very much from the Kuzikyo.
I could see the question.
Yes. Okay.
Yeah, it says, essentially the question is, are there any other chemicals in the ultrafine particulate matter that we used in the exposure experiments, like metals or something like that? No, we don't. You can put metals into using this system. We just didn't do it. We used a particulate that had been used previously by both Tony Wexler here at UC Davis as well as Laura Van Winkle. And so it was a well-characterized particle. And so that's one of the reasons we used it. Again, yeah, as far as you could say there are other substances in there, but they're below the level of detection for the analysis we used for looking at the particles. So, but there are no metals that you might see, regarding traffic-related pollutants that you would see with regard to brake wear or stuff like that.
Okay. The follow-up with the question, I'm curious. So your data in the slide 7 showed that the ozone and ultrafine particle that from the ozone react with the PAH, actually PAH contained in the combustion drive ultrafine particle, and they produce a toxic product, right? So those chemicals were really affecting, damaging the lung and heart. So do you have any idea, like which chemical specifically that is affecting? I know they have a list of chemical.
Yeah, that's a good question, actually.
Yes.
What we're seeing here is that when you look at this and you look at the data, what we're seeing as a question, is this particle or the composition of this particle with regard to the combined exposure more toxic than without ozone? And I don't think we could really answer that question with our data. When we look at the ultrafine particle response by themselves, especially with regard to microthrombi, I mean, we still have a response. And with regard to injury to the myocardium, we still see a response. Now, it is greater with the combined exposure, but is that due to the fact that ozone may be increasing the permeability of the airway and increasing the uptake of the particulate matter into the bloodstream? So, there's questions there that would need to be addressed. We've thought about, doing studies where we take the UFPM plus ozone, remove the ozone, and look at the UFPM by itself. That was treated with the ozone. So are these as a particle composed of this material more toxic? We haven't done that study. That's technically difficult to do, but Tony Wexler has told us that we could do it.
That'll be great.
But it would be, a lot of technology there that we would need to develop to do that.
Right. It is very important study because it's we are thinking that just ultrafine particle alone and ozone alone, that's not that way work. So they are combined and react and then produce a more toxic, harmful, chemical producing. That's So we need to study.
Yeah, we would need to do another study on that.
Exactly. Thank you. Yes.
Another
question.
Yeah, we have another question. Would you consider the proposed injury mechanism to be more relevant to chronic repeated exposure to normal urban air pollution levels, or instead to unusually high acute event exposures? That is a good question. Well, first of all, with regard to the PM, we did do, and in Wong 2018, we did do modeling of particulate exposure with our exposure levels that we use, and compared using deposition models that are available in the literature, both for rodents and for humans. And the equivalent dose for a human would have been a high ambient acute exposure. So similar that you might see and has been measured roadside in LA, for instance. Close to the roadway levels of ultrafine particulate matters, these have been seen. Now, if you go to other international cities, this would actually be well below the exposures that you would see in cities like, Beijing or other cities in the world where ultrafine particulate matters could be quite high. With regard to the ozone is, again, historically been picked based upon comparison of dose response in rodents compared to humans and the type of essentially neutrophil influxes that you see with the comparable exposure to around high ambient levels around 0.2 parts per million or 200 parts per billion. So relatively high. So yeah, they are on the high level, but when we designed the initial experiment and study, we wanted to know that we were in a range, that we were on a higher level of exposure, and say, okay, this is our high level. Do we see effects at this high level? All right, next question. In humans, what kind of impacts might you see from the thrombotic effects, fibrosis development, arterial blockages? Well, what you would see with these micro, we're not seeing large, we're not seeing large thrombi occluding, right, large coronary arteries that would cause a cardiac event in this model. These animals don't necessarily have coronary arterial blockages. They have changes that are associated with chronic hypertension. They do have damage to the myocardium associated with that. And so in a human model, this would be something more associated with chronic hypertension and the changes you would see with chronic hypertension. But in an individual with full-blown cardiovascular disease, which is more typical in humans, where you would also see arterial sclerosis, these types of lesions may contribute to arterial blockage in large arteries in the heart, but would also lead to these small areas of ischemia within the myocardium, and they can contribute to the development of arrhythmias because that could influence the path of depolarization and electrical signals within the myocardium. So what we would expect to see with this type of occlusion is small local ischemia leading to the types of lesions we saw in our rat model, and the acute consequence that we would see in a patient, or that you might expect to see in a cardiovascular, somebody with cardiovascular disease, would be maybe an increase in, which we also saw, an increase in arrhythmias, and some of them being quite severe. We saw some severe arrhythmias in these animals. Matter of fact, we had one rat that went into AV block, and essentially its heart stopped for a minute. So, it was really weird to look at that EKG, but it's very severe AV block and very severe PVCs as well.
Okay. Another.
Okay. What is the basis for ultrafine particulate matter speciation? Okay, so maybe I need an explanation what you mean by speciation.
Arash, could you unmute Bart so that he can ask the question?
Sure. If they can raise their hand so I can, okay.
Yeah, okay, right, okay, thanks.
Hi there.
Hi.
Yeah, no, so the ultrafine particulate matter is representative of what sort of process or air sampled stream? Why did we choose these particular ones besides they were available?
Well, the particulates in the composition is very similar to diesel exhaust. So that's one of the justifications for and why we used it. And the reason the system for producing these particles was originally produced in the first place. So that's the justification based upon environmental exposure. It does replicate diesel exhaust relatively well as far as its compositions, especially with naphthalene and other components. With regard to another reasons why we specifically use this exposure system is because unlike using concentrated, particulate matter from the environment or using, collected particles and then resuspending them and using intratracheal installation, is that one is an inhalation. This is an inhalation exposure. It's to an exposure level that for a rodent is very close to what we would see in a human in high ambient exposure. And we can produce this atmosphere is highly reproducible. Every time we turn on the equipment and we set the burner and the flow rates through the burner at a certain rate, we get a very consistent particulate and its composition is very consistent. Is ultrafine particulate matter equivalent to PM 2.0? I believe that, but I hope that answers your question.
Well, it certainly helps me very much to understand why you chose this, and it's great for studies to be able to have a consistent stream, certainly. But the fact that the particulate matter reacted pretty quickly with the ozone would bring to question whether, unless someone was sitting right at the exhaust of, like school kids sitting next to the exhaust pipe, which I always hate. But more generally, if you're just next to the freeway, wouldn't these this process stream, this exhaust stream, wouldn't it have had plenty of time to react with oxygen in general, not just O3?
Yeah, I would agree with that. I think that it was one of the observations we had, which we were not necessarily expecting to see, that really indicated that, when we look and we talk about PM or ultrafine, any type of PM within the environment, the question is, what is it co-exposed with? And what is the co-pollutant mix? It's not just, and as we all know, particulates as a term is not like looking at a gaseous pollutant. It's not like talking about ozone. It's so variable in its composition, its size, and which means deposition, chemistry, all these things are different when we go from one environment to the next. So it's really critical, I think, when we talk about particulate matter, when we try to start understanding maybe on the more mechanistic level of what's driving biological responses, to really ask, well, how does this change in composition influence toxicity and biological response? So yeah, I agree 100%. This was kind of something we looked at and said, okay, so if you go and you look at, near roadway during the winter when oxidant exposure or oxidants might be lower versus during the summer, or when we have high sunlight, high temperatures, when you might have oxidant pollutants such as ozone present, does that change the characteristic and the toxicity of that particle, even though it's coming from, as far as an exhaust, from the same source?
Right, and those are great questions, and we can't look at all questions in the universe, but certainly my takeaway is that for some pretty ugly things, there is an exposure pathway directly to the myocardium. That's right. Interesting. That's very interesting. Thank you very much. You're welcome.
Another question is UFPM equivalent to PM 2.5? It's actually equivalent to PM 0.1. So ultrafine particulate matter is a subset of PM 2.5. It's not generally measured. When we do an atmospheric measurement of particulates, we generally break it in PM 10 and PM 2.5, which is a smaller component of that. But when we actually look at the number in a typical, when we look at PM 2.5 levels, quite often the number of particles that are below 0.1 micron are actually the most of the particles, can make up. So for instance, in exhaust, there is PM 2.5, but the majority of the particles, especially out of, unless you have your burning oil in your engine, most of the particles are coming out as very small ultrafine particles, which are smaller than 100.1 microns or 100 nanometers. And the particles we used were around 70 nanometers in size.
Okay. One question that Heon was asking, timing is only potential explanation for oxidative stress result. Are there any other reasons?
Yeah, I mean, one would be that the oxidative stress isn't great enough, but the argument against that is that the lesions we're seeing in the lungs are severe compared. I've been looking at ozone-induced injury for my whole career, and the lesions we saw in the combined exposures in both the Wistar rats as well as the spontaneous hypertensive rats were severe injury, oxidant stress injury. So, you would think that you would be able to see if we sampled it correctly, markers of oxidant stress. Now, what we were trying to do was co-localize that stress with the particulates themselves, and the timing for that, definitely timing was important there, because letting an animal sit there for 8 hours after being exposed to a particulate, where that particulate goes becomes very important, and how much ends up remaining is going to be very small, especially for these ultrafine particulates, which can be taken up by the vasculature.
Mm-hmm.
Thank you.
I have one question that in general, ultrafine particulate matter enter the body through the lung. And then nowadays they were saying that it also can pass through the blood-brain barrier. So did you observe the translocation of ultrafine particle in the lung and heart? Did you have a chance to look at it, like particle deposition?
Yeah, again, we, our hyperspectral analysis again was, I think our timing was off. I think it's a really good question. I think it would be something worth revisiting because these particulates can be characterized in the tissue. So if they're getting translocated and staying intact, and there is a carbon core to the particulates, you should be able to track them into the tissue.
Yes. Yeah. At least black carbon or something.
Right,
Yes.
Exactly right. And so and I have seen that work where you translocation. And I'm not surprised by that, that they would translocate. And in rodents, there's also the idea that they could actually cross directly across the nasal passages because you have a very close contact with what do you call it? I forget what vascular bed that is. That's right at the base of the brain that would give access to those particles to the blood-brain barrier.
Yeah. Right. Yeah. I have one more question. So James Nakashima asking question. Interesting work. Is there any evidence that these kinds of ozone and PM co-exposure might lead to similar pathological change in humans?
Well, that's a good question. And I'm not sure that I think being a person who also has done considerable amount of human exposure studies, I think it would be extremely hard to set up a study now and put human subjects into it so that you could actually evaluate that question. I suspect that yes, you would see a an enhancement of the ozone and loan effect with particulates, or particulates plus ozone type of response. However, I think our ability to evaluate that would probably be difficult. And I think we'd have to put some creative thought into how we would do that. Whether we're looking at, functional studies, and where you do have combined exposures, such as, where you might be able to use, population exposures in the environment, or taking individuals that on specific types of days and looking at their responses and longitudinally and characterizing the pollutant exposure and getting measures of systemic inflammation, as well as markers of lung inflammation, and trying to see whether there is some type of relationship between combined pollutants. And then you could use as a standard a controlled human exposure to ozone and see if there's a difference. So trying to match the ozone exposure that you see in the environment with the exposure that you would see with combined exposure. And that would be my way I would think about going about doing that. But again, that's a labor of love there, getting that type of study done.
Yeah. I have one last question from me. What are the human year equivalent to the age of the rat that you use? I believe 50 weeks old. Am I correct?
Yeah,
So
they're 50 weeks old. The equivalence would be to a somebody between, I believe the number is 45 to 60.
Mm-hmm.
So you get above, you can't call them elderly until I think they're like almost a year and a half.
Mm-hmm. Yeah.
And so, but these would be, adult, the equivalent would be an adult human. So somebody between the age of, 40 to 60.
Yeah, right. Yeah. So yeah, we should be careful. Thank you. Yep. Any more question? So we are only left 9 minutes. So if there are any question, please raise the hand so that we can unmute the audio and ask a few more questions. Okay, maybe a last call.
So yeah. So anyway, if we're finishing up again, like I said, I've always appreciated working with the Air Resources Board, and like I said, my arc of my career has kind of started with Air Resources Board and is ending.
And how soon you're going to retire?
July 1.
Oh, July 1, Saturday. Oh, yes. Good for you. But yeah, we will miss for the sciences, we'll be missing you. You contribute.
I don't think I'm going to get it out of it totally. So.
Oh, yeah. All right. Like, I know Dr. Wilson is still there. So
yeah,
somehow.
Yeah, exactly.
Yes.
All right.
Okay. So thank you for joining us today's seminar. Thank you, Dr. Schrager, for your presentation, excellent presentation and valuable data. Lastly, if you have any further question, then please feel free to email me. If you have a question to Dr. Schrager, I will forward the question. And yeah, thank you so much, everyone. Congratulations, Dr. Schrager, for your retirement. I will keep in touch.
All right, very good.
Thank you.